please welcome President of the Humboldt Bay Mycological Society, Mandy Hackney, and her talk about the California Fungal Diversity Survey. Thanks for joining us, Mandy, and please share a little bit about yourself and your journey as a mushroom enthusiast and researcher before you get started. The floor is yours.
Hi, thanks for having me. I'm coming to you tonight from We Outland, aka Eureka. I got interested in mushrooms. Honestly, when I was a kid, I really liked Alice in Wonderland. When I came up to Humboldt, as you can see living up here, it quickly became an obsession. And I've been volunteering with the Mycological Society. I've been working for FUNDIS, and it's still just like a super big passion of mine that I've had all these years and hope to do the rest of my life. Well, thanks everybody for joining in tonight. I'm here to talk to you about FUNDIS, which is short for the Fungal Diversity Survey, which is a nonprofit organization that I worked for temporarily. They had grant funding to do this really cool survey project in the state of California. They were funded by the California Biodiversity or California Institute of Biodiversity. And all the photos you're going to see here tonight are my photos that I've taken in the field. These are all specimens that were collected. And this one right here on the opening page, if anyone's curious, is the cinnamon webcap. And I found this at Malel Dunes. And I do have two screens, so you are going to see me bounce back and forth so I can see my notes. Okay. So let's talk about Fundus. Their mission is to protect biodiversity of fungi and do this by conservation. They want everything to be visible, documented, really protected and appreciated. And they have a few ways of doing this. They've been increasing public awareness and they've been helping community scientists like myself get involved and to be able to work with people who are already in that field professionally. And this mushroom you see here in the background is a really pretty waxy cap that we have here in the redwoods that I collected from Headwaters Forest Reserve. So I first got involved with Fundus as a volunteer years ago. They have a fungal diversity database on iNaturalist, which is a project that's open to the public. Anybody can join. And I was a project curator. So basically, what I did was I would welcome new members. I would go through and I would delete low quality observations, and then I would kind of scour iNat looking for high quality observations. And just to give you an idea what I'm talking about when I say, what is a high quality observation? Over here, we have a photo of this amanita that shows mushrooms that are in all the stages. You can see the cap, you can see the gills, you can see everything about this mushroom. You can even see a little bit of the habitat with the leaf litter. We would call this a high quality observation because it hits all the points. We see everything we want to see about this mushroom. And I put a few examples over here to kind of show you what a low quality observation looks like. Something that's kind of too far back, like we can't tell what it is, or I'm pretty sure everyone has seen these where it's just a photo of the top of the mushroom. There's no other photos. That can be really difficult to identify considering that some mushrooms look so similar. We really need a gill shot. Or down here, you'll see this a lot where it's just like a blurry mushroom photo and you're like, why did you upload this? So just to give you a little idea, if you're interested in getting involved with Fundus, you absolutely can. You can join this project on iNaturalist and you can upload your high quality observations. And they have different curators now. I no longer hold that position. If you need help, you can reach out to them if you have any questions. They also offer rare fungi challenges, and they have different challenges for different areas in the country. And so I put a little picture down here to show our West Coast Rare Fungi Challenge. And you can print these on their website. They have these little pamphlets that are pretty handy and they tell you what they're looking for, where to find it, when to find it. And if you do find it, they really want you to go out, take a high quality observation, upload it to iNaturalist, and then there's people who specifically volunteer with Fundus that just do the fungi challenges, and they'll probably come through and give you more information and be like, congrats, you found something really cool. We super appreciate you like uploading this. So that's another fun way to get involved if you're like wanting something a little more of a challenge. You can go out and try and find something that's more rare. So as we continue on our Fundus journey, so Fundus has a separate project called the California Fundus Project. This is the one that was getting grant funded, and this project was the first of its kind. There was no other project like this in the world. When I started, I was the first one that got hired. It was really cool because I was already volunteering for them and I was already kind of doing this as a hobby where I was just going out and making high quality observations and collecting specimens and sending them off for sequencing. So I was like, hell yeah, I'm gonna get paid just to do my passion. This is like a dream job. And so essentially what they would do is we would get permits for different areas. We would go out and the goal was to try and get like a snapshot of like what is the fungal biodiversity in the state of California specifically. And the goal was to hopefully have this happen for 3 to 5 years. It did not last that long. I worked for them for 2 years, and then this past year they were able to operate at about 15% of the original budget, and they kept on a few people, and their focus was mainly bugs that they were finding living in mushrooms and also like soil samples, along with still collecting some fungal specimens. And all of these are added to iNaturalist and it's linked to the sequencing results. And you can get online and you can check it out. All you need is an iNat account to look at it. And so this is helpful because we're creating a baseline for the data for fungi in California because as a lot of people know, fungi gets kind of ignored. It does not have the same attention as like birds or plants. If you get on iNaturalist, it doesn't have as many observations. It doesn't have as much quality like identifications. It can be harder to identify. If you use iNaturalist, you know that sometimes it's not really good at IDing your photo because it just doesn't have the same amount of data. And so that's what we were trying to like bridge this gap of like creating more data to make it more accessible and to see what we have because it's really important to see what we have because we don't really know. Less than 5% of fungi have been documented. There's a ton of stuff out there. And when you look at other fields, that's kind of absurd that less than 5% has been documented. And so if you're super interested about the actual statistics and things that they found in this project, you can go on their website and they have year 1 and year 2 reports that you can check on for free. You can get on there and look at them. And that's about all I have to say about that issue with the project. But so
Before you go on, Mandy, we did have one question in the chat that I should have caught earlier, but they were asking about high quality pictures of fungi and how do you take them without disturbing them in their habitat?
well, so that's a good point. Some places you're not allowed to pick, even for science. There's a lot of phobia when it comes to mushrooms and some antiquated laws that should be changed about harvesting. Um, what you can do is you want to always shoot for like 3 photos. You want the top, you want the underside, and you want the stem. And if you remember to, you can also take a few feet back and you take a habitat shot, especially if you're not that familiar with the plants around you, because certain mushrooms have relationships with certain trees. So I would say, you know, if you're in a place where you're not supposed to be picking, which is a lot of places, you should just be as gentle as you can. Sometimes what I'll do is I'll take a picture of the top and the side, and then I'll like lightly lean back the mushroom and get down on the ground and like reverse my phone. Some people will use a mirror and they'll just like look at it. Um, I think it's kind of silly that we can't pick mushrooms. You can pick all the berries you want, but you can't touch the mushrooms. So maybe check in with your local area and see what their protocol is, if you're allowed to do that there. And if not, maybe just be a little stealthy about it. And when you're done, you just pitch it into the woods and you're doing the spores work because there's no waste in nature and it's going to go back to the earth. So I hope that answers your question.
Thank you.
So to look at the projects, I was called a fungi collector for this project. And you're probably wondering, that sounds really cool, Mandy. Tell us more. So I would basically in the beginning, we didn't have a lot of permits. So I was going around getting permits for a lot of places up here in Humboldt County. And some of the major permits we had through the project was like BLM, state parks, different things like that. I managed to get a lot more up here just because I was adamant that I wanted to collect more up here. And so I would basically go out into the field, bring my gear, make these high quality observations, collect my specimens. I'd bring them home. I would follow our protocol and use our tags to keep things labeled. And I would upload my photos to iNaturalist and upload them to the project and put in all my notes. And then I'd pop them in the dehydrator. wait till they're nice and dry and crispy, package them up properly, then wait till I have enough to basically pull tissue. As you can see in this picture up here in the corner, it's kind of what it looks like. So when I say pull tissue, we're talking about getting these specimens to undergo DNA sequencing. And in order to do that, you need to pull off a little teeny bit of the fertile surface. So on most mushrooms, that's the gills. And then you have these little tubes that you like pinch off a little bit of tissue and you put it in and you close it really quickly to try and keep any other contaminants getting in. And most of we'd have like little trays that would have about 90 tubes. And so I would do that, and then I'd have a spreadsheet and I'd have to type in which one I'm doing and make sure that all the data lines up. And you have to be like sterile in between and sanitizing things to try and keep it from getting contaminated results. And then once I had that, I would take my little plate and tape it up to make sure it didn't come out. And I would package up all my specimens so that they wouldn't get damaged in transit. And then I would mail them off to our sequencing team in the Bay Area. And then they would continue the process of actually doing the sequencing results to find out where these fungi that we're collecting land in the tree of life. And it was really cool to be able to do this. It was definitely the coolest job I've ever had. I will be honest with you that, that pulling tissue is super tedious, especially when you have like hundreds of specimens. Um, that was probably my least favorite part of this job. So these specimens get mailed off. Where do they go? Down to the bay at CSU East Bay. They have a fungarium there. And so basically what would happen is when they would get there, they have, if you look down here in the corner, they have this massive deep freezer and they would freeze everything upon entry just in case there was any hitchhikers, bugs, bacteria, anything that could contaminate the fungarium itself. They wanted to kill that off before it became an issue. And then they had their own way of labeling things. They would basically unpackage it. They would print off all your notes and put it in a little box that's labeled. And then these little boxes would be stored um, in a larger filing cabinet. But before they went to their final storage position, they would put them in. This is a massive, massive dehydrator. If you guys can see, it's taller than I am. It's super impressive. And they just basically want to make sure everything is super dry and crispy so that there's no mold and there's also no like bugs or anything that could hurt the rest of the specimens. And then they get stored away in these little neat cabinets and you can go and pull them out as needed. My friend Warren, who is a collector for the project as well, was running the fungarium at the time. And I was lucky enough to be able to go down to the bay and get a tour and take some photos and see it firsthand. So it was nice that living in California and not too far away, I was able to get like the full gamut of the project itself. So, what did I find? Where did I go? I bet there's some people in here tonight that can recognize this photo. This is the Lost Coast, one of my favorite places on the planet. For people who aren't from the area, the ocean is back here, and we got a ton of coastal fog rolling in, and it's just a really gorgeous place. And this is my home, and so this is where I made the majority of my collections, which is why I'm going to talk about it first because there's a lot on here. In that photo, it is also Paradise Ridge Road, if anyone down in Sohum is curious. So here's some of the things that I found that came back with interesting results, or they were just like, I love them, or they're pretty. So I was like, I'll throw them in here. In the upper corner here, we have this Mycena austera, which came back, and it's the second observation on iNaturalist in the world. And all the collections I'm talking about tonight, I'm going to be mentioning just on iNaturalist. This doesn't mean that they haven't been found before. There are other like microweb portals and other places, but everything I'm referring to tonight is from iNaturalist. All these were collected by me. All the photos are by me. And this one's really interesting because it came back to be something super uncommon. And this is my backyard. I would just go in my backyard and collect specimens and photograph things. And then sometimes it comes back something really cool, which was really fun. And up here we have the parrot mushroom, which a lot of you have probably seen if you go walking in the redwoods. This is one of my favorite mushrooms. It's absolutely gorgeous. It can come in different shades. It can be anywhere from green to yellow to pink to blue. And they're very slimy. I absolutely love them. If you haven't seen one, go take a walk in the redwoods right now because I was out there yesterday and I was seeing them. And this is at Headwaters Forest Reserve, if anyone's ever been there. It's a really pristine redwood forest that we are lucky to have up here that was saved from some logging and it has old growth in it. There is a correlation with fungi and old growth. There's certain things that only grow there. And it's a really magical place. I love it. I don't live far from it. And this is another thing I found down there. This Mycena terrigina in this bottom corner, you can see these are really small mushrooms. And if in case you're wondering what these grow on, that's a rotting lady fern. These are really small and they're kind of pinkish to white and they're just really cute. And it's like a super fun thing to do when you're out with people to be like, let me show you this mushroom that grows on this rotting fern and give you a chance to like find it. So putting that on your radar, go check your lady ferns. And down here in the corner, I have this gorgeous blue Antiloma that I also found in Headwaters. And I just love this specimen because if you look at the gills, they're what we call marginate, and they have these like coloration on the edges of the gills themselves, which I think is just absolutely stunning. I love blue fungi. Blue is like a color we don't see a whole lot in nature, so when I find it, it definitely gets all of my attention. And keep going here. So in the upper corner here, we have another waxy cap that is the Gliophorus. This is Irrigatus CAO1. And basically, when you see things with like kind of mouthful names like that, it means that it's come back as a different species, and that's kind of like a temporary name. I also found these at Headwaters Forest Reserve, and this is a really cool collection because it is being used in a publication for Gliophorus sect unguinosae. So I am helping to co-author this paper because they are using one of my specimens that I collected here locally for the project. And that's really cool because what's funny is I found these like right across from the vault toilet at Headwaters. It's like you never know where fungi is going to be. And over here in the upper right, this is a really stunning photo. This is one of my favorites. Also, I have a lot of favorites. You're going to hear me say that. But this is definitely a fave. If any of you came to the fair this year, you saw me wearing a hat just like this mushroom. So we locally have a mushroom called Hidnellum peckii that is strawberries and cream or like the devil's tooth. And so that's what I thought this was when I collected it. And when it came back, it was a different species. It's the Diabolus PNW06. And I collected this up at the Redwood National and State Parks. It is gorgeous. This mushroom creates this guttation, which is a fancy word for droplets. So you see this droplets coming out of it, and it has this compound in it that colors it red. And this was also a really unique specimen because if you look at this photo over here, there's some malformation on top, which I had never seen before. This mushroom has what we call teeth or spines on the underside, and they're not normally exposed. So this was like just a really cool treat to find this kind of monster looking mushroom with this malformation on top. And I just love showing this one off. Down here in the middle, I found this truffle at Headwaters Forest Reserve, Tuber pacificum. It is the first record on iNaturalist in the world. This thing was super tiny. If you look over here at this photo, these are our tags we would use to keep things organized and also for scale. This thing was super small, and I only found it because I was out with my colleague, Adi, and she had come up for a visit, and Adi was photographing a gibellula, which is a species of fungus that parasitizes spiders. So while she was just taking her time photographing, I was just going around and flipping over logs like one does. And I got lucky and found a rare truffle. So, yay. I'm super excited that we have truffles. Truffles, again, one of my favorites. I know I say that a lot, but it's very true. And down here in the left-hand corner, you're probably thinking, like, hey, what do you do when you're in the field for a few days? How do you take care of your stuff? Well, this picture shows it. You collect it in your tackle box, you keep it organized, you put it on your dash, and you hope for the best because sometimes you are out in the field overnight or for a few days and you don't have access to a dehydrator. Mushrooms create heat and they can sweat inside your box. So it's a really good idea at the end of the night to open your box so your stuff doesn't get all gross and start to decay faster. And especially, it's really handy for things like inky caps, which are mushrooms that are really fragile and have a tendency to not last very long because they deliquesce. And deliquesce is just a fancy word that means to turn from a solid into a liquid because they literally just become goop and people will use it for ink. So I always tell people if you have something that's very fragile and goes very quickly, like inkies, immediately put it on your dash. Don't even leave it in your tackle box. And if you're out overnight in the field, open your boxes up at night, put them on the dash, trying to get them to dry a little bit is the best way to go about it. Or if you're lucky, maybe get like a homie that can splurge on an Airbnb and you guys can plug in a dehydrator. This is really cool in the upper corner here. This is basically any truffle that is not in the genus tuber, which is the super yummy edible truffles. They call it like a false truffle. But this one's really unique because it's a russula truffle. And a lot of you are probably familiar with russulas. They're normally like, medium-sized mushrooms that are really brittle. And so we jokingly call these truffulas. It's like a combination of a truffle and a russula. If you look at the gill system here, you can tell that this was just like a mushroom that once, that would grow above the ground and had gills. And then somehow along the line, it eventually became a hypogeous fungus, which hypogeous is a way to describe mushrooms that grow under the ground. And so this one was a rumpant, which is a fancy word just saying it was popping up through the dirt. So I saw it sticking up and then I cut it in half and I was like, oh, this is really cool. And some of these too, if you find there's like different lactarius, which is like a milk cap, sometimes they make truffles and you can squeeze them and milk comes out. But I found that at McKay. I got a permit over there and it's close to where I live and it's also a magical place where I find cool things and I love going there. In the upper corner here, we have this is a huckleberry leaf. And if anyone is familiar with our evergreen huckleberry, it's not very big. I found this at Headwaters Forest Reserve and I adore it. The fungus has created this kind of design on the leaf, and you've got these tiny little fruiting bodies, and it was just absolutely stunning. And if you even look closer, some of the fruiting bodies have like water droplets on it. And I put a photo down here below it with one of our tags just to show you like how small this was. Because I love tiny things and I love finding them and photographing them. It's really fun and it's like a challenge. And also to get access to this part of Headwaters, there's a couple different ways to get into Headwaters. One is the Elk River Trail, which is open to everyone. Then they have kind of back entrance, which is down in Fortuna, which is the Salmon Pass Trail, which you need permission and a gate code to get in. And then there's a third one that most people don't really know about, which is a backcountry entrance, which is on the way to the Elk River Trail. And basically, for me to get access to that part of the forest, I had to get a CB radio installed in my truck because it's in an active logging zone. So I had to like get special permission, get a special key, spend a couple $100 that Fundus paid for getting a CB radio installed in my truck so I can go in this backcountry area that's only accessible with a 4x4 high clearance vehicle. And because it's pretty dangerous because you're going back there and there's like huge logging trucks and there's roads that are like overhanging like the edge. And it's really awesome because there's like pristine old growth redwoods that nobody else is there and it's really magical. And so it was totally worth it. Um, and that species, the Capitotricha cao2, another mouthful, is a new one. This was a new name. I found it. They put a new name on it because we hadn't found it yet, which is also really exciting, which goes back to like, we really need to protect these places because we, for one, don't really know what's there yet. And we also know that there is this like relationship that we can find things there. And down in the bottom here, I have a picture of another waxy cap. These are kind of more slender. These were also found at Headwaters Forest Reserve. This is Hygrosabe PNW 19. My colleague Sadie, she's incredibly smart. She's super impressive when you're out in the field. She knows like everything. She's like a walking field guide. She first discovered this species of waxy cap in 2019, and I believe it was up in Washington State. So it was really cool to be able to see that it is being found elsewhere. Up here on the left, we have a picture of a deer mushroom. A lot of you probably know that we have deer mushrooms, and they're called deer mushrooms because their spores have these like little antlers and they kind of look like deer. I found this out at Wax Creek, which is up the 299. It's BLM. This mushroom is being used in a publication about Pluteus leoninus clade in the Hollow Arctic region. You can see why I had to make notes and write all this down. It's a mouthful, but it's really cool that like the things I'm finding here are being used all over the world. And we're finding that some of the same species are here and some of them aren't. And it's just really interesting. And it's also cool to see the networks and the connections and all these cool people you're getting to meet and getting to see their work that they're doing. And speaking of cool people, this is my colleague Adi. And I believe that was the Jibalula that she was photographing when we were in Headwaters the day I found the truffle. It was really cool that this job that I had which was basically just getting paid for my passion, brought people up here because we had permits all over the state. So I could basically collect anywhere in California. And a lot of my colleagues would come up here and hit me up. And it would be really fun to get to go out and like meet new people and watch them photograph and see the things that they're finding. And I really like that because there was a lot of times I was out there completely alone, a lot of times just hiking alone by myself. And, anyone who identifies as a woman knows that that's not always like the best because the world can be a scary place. And so you kind of have to work up to like being able to push yourself to get through situations and outside of your comfort zone to be able to like go and collect and find the things that you want to find. Over here, we have another photo of a mushroom that I found in headwaters. As you can see, these are pretty small. This is Atheniella aurantiadisca, the tangerine bonnet. I think they're gorgeous. And this is redwood duff. So these are really small, just little tiny babes that really pop against the redwood that I just love finding little things. And speaking of little things, down here in the middle, these are Mycena monticola that I went up to Horse Mountain, and I got lucky that when I went to Horse Mountain, there was snow. And so it was just a really gorgeous day for photos. These are one of my favorites, which yet again, you're going to hear me say in the upper corner, the Geastrum floriforme, Earth stars. Well, we got dune stars, and they are that small all the time. They're super cute. If you look it up on INAT, it's going to say the Daisy Earth star, which I think is just a name. So I call them dune stars. Feel free to use that name. This is my colleague, Andrew. This was his birthday. We were down on the South Spit where we were finding these, and they have a box full of them. And over here in the upper right, I have a picture of another truffle. This is the fall Oregon white truffle, which is like a yummy, delicious edible. And I found it on Christmas in Sequoia Park. It was also rumpant. And as you can see in the photo below, my nails totally match. I've got Christmas nails with my Christmas truffle. It was a good day. I think these are really pretty. And this is another thing that I found in headwaters. This is a type of fungus that parasitizes insect. It's Samsoniella. And this was at headwaters. And I put a photo down here on the left just so you can see how tiny these things are. These are really small. These are insects. And the only thing that was visible, if you look right here, was this little puff of white. And then I was just like, that looks fungal. So then I like pulled out my hand lens and I'm like using my tweezers and going back and forth because I'm farsighted now. So it's kind of difficult to like look at little things without the assistance of technology. But I got a really cool photo. And it's like a different species I'd never heard of. So it was really cool getting to like expand my knowledge working on this project. Down here is one of my faves. I'm probably just going to say that every slide. But birds' nests are so cool. When you do a cross-section photo like I did here, you can see they have these little egg-looking things that are spore packets, and they're encased in this thick gel. I love always taking a cross-section just so you can see how they look on the inside, and they'll eventually open up and it kind of looks like a little nest. Up here is the Arthroderma CAO1 that I found in the dunes. This is a new species that I found, and they created this name for it, and it was just on an owl pellet on the ground, and it kind of didn't really look like much. But I also love owls, and I know that there's certain fungi that will grow on owl pellets, so I was just like, this looks weird. I'm gonna grab it, and then it was something different, so that was pretty cool. And down here in the bottom right, we have Ophiocordyceps variabilis that I found in McKay Community Forest. This is another fungus that parasitizes insects. This is actually a fly larvae, and I found this in wood, and it's another situation where you kind of got to get down and pull out your tweezers and try and get it out without destroying it. Ooh, let's go back. Okay, sorry, flipping through everything. Okay, down here we have Xylaria bulbosa, CIO1. As you guys are probably pretty familiar with our area. We have Xylaria hypoxylon, the candle snuff fungus, and then we have Xylaria apiculata, and this is our third species. This thing is kind of wild. It is the first record on iNaturalist for North America, and if you can see down here, this is a bulb. Like I had to dig this thing out, and then I cut it in half, and it's like firm and like white on the inside, and I was just like, what is this? Really cool find, blew my mind. I didn't even know we had another Xylaria, but it's there. This is a really cool one. Up here, we have another Ophiocordyceps species. These grow in the dunes here locally and up through Oregon. My friend Connor Dooley actually found these when we were out on a trip. He's a very talented mycologist who's excellent at ID. We actually have him come and he works our ID table at the fair. These are June beetle larvae or June bugs. June bug, June beetle. Can't remember. But they basically, these larvae like to feed on the roots of the beach grass and then they get parasitized by this fungus. And if you see these kind of black sticks sticking out of them, these are what's called a stroma. And this is like the part of the fungus. If you were to cut these open, they're like black on the inside. They've been completely eaten up by fungus. And what you're looking for is like right here in the middle, is basically a needle in a haystack. You're looking for something that looks like a stick. And I put a picture of the habitat down here, just how you can see like how challenging sometimes it can be to find these things because you're digging around in sand and grass and you're looking for something so little. But when you do get the knack for it and you do have the eye for it, it's really fun. And I highly recommend y'all get out there because I just found these in our dunes like a couple weeks ago. So they are there. Um, and we're flipping. Okay. In the upper corner here is another species that parasitizes insects. Well, this one specifically spiders. This is what I was talking about earlier. It's Gibalula, which is really fun to say. I found this. This is actually one of the biggest ones I've ever found. It was on a stick on the ground, and I put a photo down here for you to see what it looks like. I use a TG, an Olympus TG6, if anyone's wondering what camera I shoot with. They're really small. They're like waterproof, tough outdoor cameras. Highly recommend some lighting and a tripod if you're going to use it. It's just kind of like right here. You can barely see in this photo of the Gibalula, but those are so cool. And over here, this is my big mushroom crush, is Catanella olivacea, also known as the olive salver. Stunning. These are stunning mushrooms, as you can see. And what's fun about them is in the photo right here, when you put a UV light on them, a black light, they fluoresce this bright orange. It's really stunning. Highly recommend if you don't have a UV light, get one. And if you can, splurge and get a better one, like a $40 one on Amazon, like 365 NM is what I use. You won't regret it. It's super fun for night hikes. And it's also really cool data when you're collecting in the field because we would collect scents, we would do taste, we would do UV, we would do KOH. You know, what trees is it near? Like you want all of that. All of it is data. And even if it doesn't fluoresce, that's data. Down here
So
we have,
there's a couple things in the chat that I figure I'll maybe jump in if that's
yes,
okay.
yes.
So one relates to the TG6. There was a comment from Joe who says, look at getting a TG6 or 7, and they take amazing macro shots. So that just seemed like a relevant spot to kind of echo what you were saying about the what you're using. And then there was a question also about the owl pellets looking like they're on small bones. I wondered that too. Is that what we're seeing there, or is that something else in
Yeah.
that photo?
Yeah, because owls, a lot of their diet is rodents,
I
right? And
see.
then they have to vom up the bones that they can't digest. So that is, when you go look for owl pellets, it's normally like fur and bone, and it was growing on it.
Fascinating. That's all.
Okay, cool. Um, this photo down here on the bottom is Repartites odorous CA01. It kind of had like a mild sweet odor. And it's a new name that was created for the species. It was the first observation on iNaturalist for Humboldt County. And this species is close to one from Italy. How did it get here? We don't know, but we know it's here now. Okay. These black cluster of cups don't look like much, but they're actually pretty cool. They're an indicator for healthy old growth forest. I found it at McKay. And this Diplocarpa species, it's the first for Humboldt County on iNaturalist, and it's the second for California. So it's pretty uncommon. And up here in the right, I have another cordyceps, one of my favorites, obviously. They are parasitizing some sort of insect pupa. And this is what it kind of looks like in the ground. I put a photo here. I basically was stopping for a russula that I wanted to collect. And so I threw my pack on the ground and I got down and was pulling out my gear. And I kind of just noticed this and I looked over and I was like, oh my God, that's a cordyceps. And then I was finding them all over the place in the ground, in leaves. And I found like 20 of them that day. It was amazing. I love them. You can see the fungus is just erupting from it. It's a really cool thing to see. And this is another cool one. This is Asterophora lycoprodoides, the powdery piggyback. They parasitized rotting russulas. So you can see in this photo over here, we've got this blackening russula and it's just completely covered. This was like the biggest one I had found. It's like fit in my whole hand. It was just gorgeous to see. And I love this photo because you get to see a little bit of everything. There's like some pins and then there's the mushrooms and then there's the rotting part. So always check some rotting rustlage. You don't know what you're going to find. So this photo down here in the corner are some inky caps. It's the Narcissia. This is actually being used in a publication about Narcissia delicata that exists in Europe. So that was something cool that I got to be a part of. And this was on one of our club members' property in Fortuna. She let me come down and survey. And up above it, I have a photo of my colleague Dean when we were out in the dunes. The willow thickets are actually really undersurveyed and they have a lot of cool things there. But as you can see, it's very thick. And it was probably like the worst place I ever surveyed just because it was so frustrating because it's like flooded and thick and you're trying to get in there and set up equipment and things are poking you and you can't turn around and it's like, but there's really cool stuff. So if you fill up for it, check your willow thickets. This is one of the things I found in the willow thickets. This is Cortinarius oliginosus. It's called the marsh web cap. This is in Malel, and it is the first observation on INAT in Humboldt, and it's the second in California. The bottom here, you guys are probably pretty familiar with hedgehogs. They have teeth. They're kind of this peachy color. They're like a fabulous edible. This one I found in that backcountry entrance to headwaters. And this is a new species. They created this name SPA CaO2. And so that was really exciting. Another cool find from that area. This is a gorgeous mushroom, this blue one up here, the little blue polypore. This is part of it's on one of the West Coast Rare Challenge on that fundus list that I was talking about earlier. I found this also at the backcountry entrance at Headwaters because it's like really pristine and there's old growth in it. It was just a huge fruiting. I couldn't believe it. Absolutely stunning mushroom. If you're wondering in this photo, this little bit of red, that's the KOH reaction. KOH is potassium hydroxide. It's basically like a liquid substance that comes in a little bottle and you can squeeze it on mushroom tissue and sometimes it'll cause a reaction and sometimes it won't, and that's data. Over here on the right, we have a photo of Tilipocladium ophioglossoides, which is basically a truffle that's being parasitized by a fungus that's in the Cordycipitaceae. As you can see, this is what it looks like above ground, just kind of these little truffle clubs. And then when you dig it up, here's the truffle that it's parasitizing. I believe it's a deer truffle, or I think that's the common name. It's a Lophomyces. This was up at Sumeg, and you can see behind me kind of the usual stuff I would use. I bring like a tackle box and my basket and knife. And if you have a friend that you like playing around on walkies, walkies are really fun. Down here we have a photo of Pilobolus, which is a really fun mushroom because they're super small. You can see I put a photo here just so you can see the scale. This is on horse dung. These mushrooms are interesting because they have this body that fills with water and it gets heated by the sun. And then once it gets so hot, it literally shoots like a cannonball, that little black spore packet on top. And then that's how it spreads its spores. It's pretty interesting. So this Lacarius species up here in the corner is a little unusual. It's got this kind of raggedy stipe. This was a new species that I found in the backcountry headwaters part. And this one down here is the Entoloma anodynum, which is the first record on iNaturalist. And this was kind of cool because I found it at the South Spit and it was like the end of the day and I was getting really tired and I was like, I don't know, I'm about to leave, but I'll just walk around this field. And then I happened to find something rare. And of course, I didn't bother to pull out my camera that day because, like I said, I was tired, but those are the best photos I got. So it's really interesting that we have things that aren't documented and are uncommon and, just here in our little part of the world. This is my friend Adrian. This is when we went up to Jedediah Smith State Park, which is up in Crescent City, up near Crescent City in Del Norte County, which is like an hour and a half from here. If anyone isn't from the area, it's a really pretty area. As you can see, I put a photo of the redwoods just in case somebody hasn't been here. They're really big trees. It's a super pristine forest. It was really fun to get out there and have access to these parts because these areas normally don't allow collecting. So it was like really cool to be able to be a part of this project where I was able to go into places that no one else can go and I can collect things. And I was really, really hoping the whole time that someone would ask for my permit just so I could show them, and no one ever did. I was so disappointed. I was ready though. And this mushroom over here is really cool because it is also a new species that I found in McKay. And I put a photo over here on the right. This is the UV fluorescence reaction that it's having. You can see down here, it's got a red KOH. It's kind of just like this little brown mushroom, nothing super spectacular, but then the UV reaction was quite stunning, and I really like that one. These are really beautiful. The Jaconica horizontalis. I found these in the dunes. This is like a stunning cluster. These are really small. A lot of Jaconicas, I don't have a photo here, but they have a red UV reaction. So that's one way you can like quickly identify, is this a Jaconica? This is my colleague Tay up at SUMEG. I was taking a photo of her working. You might have seen her. She's a talented mycologist and she's pretty involved in the mycological world in California. This Cortinarius here is also under UV. It's having a really cool bright yellow UV reaction, and it's the first observation on iNaturalist for California. Over here, I put a photo of Porpulomopsis, which is the pink wax cap. If anyone came to the fair, this was our fair t-shirt design this year, which is pretty interesting. And I also collected, I was the only one on the team who was lucky enough to collect Porpulomopsis, I believe, just because I live up here and I live in the Redwoods and it's super common up here for us anyway. And all of them were kind of coming back as this, CAO1 species. And I actually collected one in Sequoia Park that came back totally different. And so we're really not quite sure. I don't have a photo of it. It looks very similar, but I just went back this week here, like, 2 years later and found another specimen. And we're going to run it again just to see, like, hey, was this like a misread? We don't think so. But like having sometimes multiple collections for uncommon things can be really handy. Okay. This um, was on the Lost Coast. So some of you down there know the area down near Nautilus and Chemise Mountain. I went out there collecting. This photo here is with Lost Coast Mike. He's a really good mushroomer who lives down there. And my friend Adrian. This photo is kind of comical. It doesn't really show it that well, but as you know, if you live in Soham, the hills are pretty steep. And I had found these cups up here in the corner, these unique looking tarzettas, which came back as, SPA CO3. We were trying not to fall down this hillside, trying to take photos. It was really a team effort that day. As you can see, I had multiple field assistants there holding lighting for me while I'm like trying to hold my camera, where we're like sliding down this hill. It was a team effort and it was just a really funny moment. And so we were trying to capture it with a selfie. This Crepidotus here in the middle is the one of three observations on iNaturalist in the state of California. These are pretty cute. They're smaller. They have a white top, and honestly don't look like much until you flip them over, and then they're pretty stunning. Over here we have the violet hedgehog, and this one is the Hydnellum CAO1, and this one has teeth. It's dentate. Really pretty mushroom. I don't know if you saw the photo I was using for this lecture on IG, but I'm like holding it. It's such a pretty mushroom. And so that's kind of like the main things that I found in Humboldt County. But I did, like I said, I had permits all over the state. So it's like, where else did I go? Well, we do an annual backcountry camp out with HBMS. So I went to the Marble Mountains. And this was our group photo for that day. You might recognize it, the Salmon Outpost. We didn't find a whole lot. It's summer, it's the mountains, it's kind of dry, it's hot, but it's still good to go and check these places out because that's still data. And you do find things. This Foliota was interesting because it kind of smelled like root beer, which I thought was pretty cool. Don't forget to smell your mushrooms. That's really important. There's some really cute little cups that were popping up over there, some Geopixus. This is my colleague Warren. He's the one that runs the Fungarium. And it was a really beautiful place up there. And as you can see, I put a photo down here. Everyone's having a good time. Everyone's keeping themselves busy. I love snapping candid photos when people are looking at things. I think they're kind of comical and it really captures the moment. In the middle here, you'll see we found this mushroom, Gymnopilus luteofolius, and it was super fluorescent. So we waited till night to bust out our UV lights and our cameras to try and get a better shot. And this photo over here kind of cracks me up because Scott Davison is in the background. He's a former president and a super talented mycologist who's been doing surveys for like probably longer than I've been alive. And it just cracks me up because he's found a really cool log and then I've found a really cool log. And so if you're wondering like what does it look like behind the scenes, it's a bunch of laying around and tearing apart logs sometimes, especially when you're in areas when there's not a lot. Another place that I went to was the Warner Mountains, which is pretty far away. It's like almost 7 hours. It's like close to Nevada, but it was really gorgeous and super far out there. My friend Warren told me about it, and I was like, well, I just gotta go. We didn't find a whole lot either. You know, it's you're in the mountains, it's really hot. Sometimes you can go to the mountains and get lucky, and there's still snow on the ground, and you have snowmelt mushrooms that are mushrooms that only grow in snowmelt. I did find this little another inky cap, this narcissia that was growing in like a meadow. This Entoloma holoconatum, this little cone bell. These were pretty. I think I just found these like where we were camping. They're sometimes you get lucky and they're right in your campground. And then this is another Cortinarius ossi that has a really cool UV reaction. I also found some puffballs. They're a little different in the mountains. This one was super warty. I found these cute little lacnolulas. They're so pretty. And then this is a photo of me up in a prairie, just like I think I was collecting poo fungi at the time, but it looks like, you know, just so charming and wonderful. And I adore that photo because it was good times. So the Clitosobe glacialis is a snowmelt mushroom, and you can see mushrooms create heat. You can see underneath it melts a little bit of it. And these will only be found where there's snow melt. So that's something fun to do if you're looking for a new hobby. Go to the mountains in the summer. I found this really pretty Crepidotus. Their common name is the oysterlings. And then this was my tackle box to kind of show you some of the things I was finding. Like I said, it was hot. It was pretty dry a lot of places. And I didn't make a whole lot of collections, but it was still really nice to go and be in the backcountry. Another place that we went to was the Klamath Mountains, which are nearby, and I'm sure a lot of you have been to, and it's like a magical eco-region. I adore it. But unfortunately, it experienced fire like a lot of places in California, and as you can see, it was pretty burnt up. We got lucky and kind of found this like little like boggy area to poke around in. There was a lot of frogs, but not a lot of mushrooms up here. It was super hot, super dry, and the river was like raging high to where you couldn't even get in it. So I found little things. This Chlorociboria, which is a green wood cup, pretty small. This one's kind of cool because if you look closely, right here, it has this malformation, which we call a rose comb, which you're not quite sure what causes that. Um, it's basically like a growth on top of the mushroom. And these are really small. I put a photo here with the tag just to show you scale of like the things that I'm looking for. And here's a photo over in the corner that shows like it was so hot and dry and we were kind of out there and everything was burnt that I was just looking for anything wet, anywhere I could collect. And I found like basically this tiny little like sliver of like water runoff that was like alongside the road. And I would just set up shop and start pulling out logs and looking for like basically anything, always hoping for cordyceps, but I didn't find any up there. And this was one of the little things I pulled off the log, this little cut fungus. Some things were too small. Like I still would go and get photos and collect them. And then some things, once you dry them up, they kind of like disappear if they're super small and you can't get them sequenced. And there also was a little bit of an issue with some of our specimens getting lost, unfortunately, when you would mail them in. I don't know what happened, but some things did not get sequenced, which was super heartbreaking.
Hey, before you move on, we had one question about the fire and wondering how post fire affects mushroom frequency and growth.
I think it varies on the species because obviously fire can be really good for forests if it's done correctly. This was not done correctly, and there are some there are some fungi that only come out after a burn. They're called like pyronicholas fungi. There's like a name for it. So in some respects, fire is really cool because it will kind of activate these dormant species that will come up after fires, and which is really cool. And then in other ways, it's like, okay, it just destroyed this habitat and things that are normally able to just thrive there cannot. So it's like, you know, this double-edged sword of like good and bad, I would say for fungi. It just really depends on the species. We also found when I was up there this little Lacaria. Some of you might be familiar with Lacaria that we have in our area. This one was pretty small and it came back as CaO4. Maybe one day they'll get cooler names, but right now I think there's so much coming in that just giving it like a state and a number is kind of like their provisional names that they're using right now. Some of you probably know Scutolinias, eyelash cut fungi. I like to call them scoot scoots because they're cute and they're little and they're really photogenic. And this was in that kind of boggy area photo that I showed you earlier. I tend to find these almost everywhere. And I think that they're really pretty to photograph because most people don't look at them, I feel like, unless you know about them. And even though we were going out there and we weren't finding a whole lot, and you're putting in all this effort to like prepare to be gone for days and downloading your maps and getting your gear and making sure everything is charged and doing all the protocol for the permits. And then you get out there and you collect like five things. So sometimes that's just how it goes, but you still make the best of it. And I just threw in a photo of our campsite and we make it really cozy and put up some lights and bring some fun things to do. And we get a fire going. And I just like camping in general. So I was super stoked to be out there surveying, even if we didn't find anything. But I always try to like mention to people, like, be a little bit flexible because sometimes people have their heart set on things. And then when you don't find it, you're like super disappointed. Another place that I went to was the Cascade Range. This is around Mount Shasta. There is a mushroom festival there every year in McLeod, and a bunch of nerdy mushroom folks get together and we camp out for the weekend. And we specifically, there's a bunch of edibles, but we also really like snowmelt fungi, which exist around Mount Shasta, which is pretty cool because there's only a couple states where there are snowmelt fungi. So California, yet again, is like a super magical place for mushrooms. And these are my colleagues, Grace, Warren, and Dean. We were all out there just collecting and having fun and doing mothing at night and UV walks. And it's just a really cool time to like get together and knowing that like this is your job. You're getting paid to like be out in the woods. Some of the things I found when I was up there, this weird looking kind of jelly-like but firm black mushroom, like gumdrop looking thing on wood, and it came back as Gadronia and a CA01. And this is like the second observation of Gadronia on iNaturalist in the state of California. And these are super small, and it was growing around snowmelt, and it's just I've never seen anything like it. They also have up there this Dendrostilbella, the cute little like teal conifer pen. They kind of have this like jelly-like, almost like a slime mold, but I don't believe they're slime mold, but they're really stunning to find when you're like flipping over logs. And down here on the bottom, we have this Mycoalia Spa CA01. I wasn't even sure when I found this. I was like, is this like an insect egg? Is this fungal? Everyone was like, I don't know, because look down here at the tag. These things are tiny. These are really small things, and you're just sometimes it's so like alien to you. You're like, I don't even know what I'm looking at sometimes. But it came back as Mycoalia spacia 1, and it was a new species. And it's the first iNaturalist observation for North America. So it's really cool. Sometimes if you don't know, just collect it anyway. This Sathrella up here in the corner was pretty cool to find at Shasta because it was literally inside a log. As you know by now, I love a good log. And one of my favorite things to do is to tear apart logs and look for things. And so I was kind of shocked when I found like an actual cluster of like a fruit body of mushrooms. I was like, what? So that's kind of cool. You never know. Mushrooms can grow on logs and inside them, apparently. Here's a different Lacunulula that I found in a different mountain range, which is really pretty. I think the species or this genus is super stunning. They're kind of fuzzy and also really small on woods. Other than the edibles when you're in the mountains, some it's mostly small things, I feel like, when we were out there. And another tiny thing that we found was the Strobilurus diminutivus. It's on a sugar pine. As you can see with the tag over here, this thing is like almost like you can't even really see it unless somebody points it out. And it's just a super cute, teeny tiny little pink mushroom. So don't forget to check your cones, 'cause sometimes there's things there. And the final place that I went when I was working was down in Mendo. There's a couple places I went with my colleagues, Warren and Sadie. We went down and camped out for 2 nights, and this was one of the like little prairie areas that we found, and did not find a whole lot. I'm gonna be honest. It was super dry. We were down there in like April. It was just very, very dry. There wasn't a whole lot out. We found a bunch of little things. One of the things we did find, which was really cool, were some truffles, as you can see down here. This is hysterangium. They were growing under manzanita. And if you're looking, it's always a good idea to like check the north slope because it tends to be a little more shaded. And so we were just kind of like digging around under manzanita and we found these and they're really cool because they're kind of like almost jelly-like on the inside and they're like blue. I had never found them before, so I was super ecstatic. We also found these chlorocyphas, the ludipes. This is on incense cedar. So yet again, very small. And I didn't know about these either, but my friend Sadie, who is incredibly talented and super knowledgeable, knew about all this stuff. And she was kind of like giving us the tour, which was nice. And I threw a photo in here of us down there. It's my truck. Sadie's over here in the hat, in the green shirt, and Warren standing here. It was a kind of a funny moment when we realized that we had all packed canned sardines as a snack. And so we each had a different brand of canned sardines. And I was like, I need to take a photo of this, one day. It'll be in a slideshow. So it was a really fun experience. And it was really cool to meet people and to collect things. And those are kind of the highlights. I threw this photo in here. This was the last day I was collecting for Fundis, which was last January. As you all know, this is the Lost Coast, Black Sands Beach, really gorgeous place. And I'm repping Fundis over here with my hoodie. So I thought I'd throw that in here. I love the Lost Coast. It's super wild, super pristine. There's no place like it on the planet. It's really magical. And I love coming down here. And that's why I was like, if I have like one weekend left, I'm going to go to the Lost Coast and collect. And now that I'm no longer working for Fundus, I am the volunteer president for the Mycological Society. So I'm still involved in the myco world, giving back to my community. We offer scholarships, workshops, all kinds of things. We have our meeting next week. Y'all should come. It's on Wednesday. And I still volunteer doing volunteer surveys for Redwood National Park. They've got a fungal survey that's been going on for like 30 some years. And I was actually up there this weekend with some friends and we were surveying and we collect data and we send it off. So there are ways to be involved. A lot of them are volunteer, but if you've got an itch to get involved, you can find a place. And I highly recommend putting yourself out there and getting involved. And so I put some contact information on here. If you want to check out the things I'm finding on my iNat account, my handle is Mandy Mushiai, or if you want to check me out on IG. I also put the website for Fundus if you want to look at their reports and see anything else that you're doing and checking out their rare fungi challenges. And then also the Humboldt Bay Mycological Society. Reach out to us, become a member. We're super affordable. We put on a really cool mushroom fair every year. If you're in the area, even if you're not in the area, come see us. It's totally worth it. Um, but that's about all I have for you tonight. That's kind of what I did, and I loved it. And I'm glad that there was some interest in getting to see what we find.
Awesome. Thanks so much, Mandy. I just allowed everybody to unmute themselves, and so we're going to move into the Q&A portion here. So if people have questions, they can either put them in the chat or they can take themselves off of mute and ask their question live or raise their hand. And I guess, while we're waiting for questions to come in, I wondered if you could speak a little bit about how certain locations are selected. And so when you're going to public land or private land holdings, how is it that you determine where you're going to survey? And is there a process as far as, how you conduct the survey within that area, like a grid that you work, or is it more just kind of, hike around and see what you can find?
That's a really good question. It is kind of just hike around and see what you can find. Every permitting process was different. Permitting was a bit of a pain because this project is unprecedented. There's never been anything like it. So a lot of times I was reaching out for permits and they're like, this is great. We want to be involved. And we've never done this before. We don't have anything. Let me type something up and we'll make something, So it's like sometimes they were literally creating the permits, like just to get us out there. And then sometimes it would be really strict, like, for state parks, you would have to highlight each trail you were going to collect on and like send them a picture. And Everyone had their own different operating protocol. Like for BLM, I'd have to like alert them every time I was going to be out there and like let them know exactly where it was going to be. So it was just kind of different for everyone, but we were trying to get permits anywhere and everywhere, especially places that we knew were super ecologically active or diverse. So like up here, I was like really interested in getting one for Sumeg and Headwaters, and I had them all over the place. I had one for CR and HSU, and I got one for up in like Hiller Park. I had so many permits.
Awesome. So we have a question in the chat, but before we go there on the diversity question, since you brought or topic since you brought that up, I'm wondering what the larger survey showed as far as our local fungal diversity compared to other places in the state and also on a national level.
So California is really unique with fungi, partly because of the species of trees we have here. We have oaks and we have redwoods, and mushrooms love both of those. And so because we have those, it gives us kind of a unique advantage as far as biodiversity with fungi. Also, Humboldt is the most mycologically active and mycologically diverse county in the state. We are very moist, we're coastal, we get fog year round, we can mushroom year round, we have unique, rare things here. We're also in a rural area, and we're lucky that it's kind of been more protected and it's more pristine. So it's kind of just a matter of like getting out there and looking. But I would say, like, if you're in Humboldt, you are in the best county to mushroom.
And would you say nationally or state-wise mostly? Or are
Both.
there other places in with around the country that would are sort of recognized as hotspots similarly?
Every state has spots. You can mushroom anywhere. It's just the season. Like on the East Coast, their season is like in the summer. Out here, ours is the fall. But because we're coastal and we get coastal fog, it gives us this advantage of being able to mushroom year round. And not only just having the coastal fog, but also we have like a Mediterranean climate. So like it doesn't really get that hot here. So it's like the mushrooms really like it here. It's really kind of like season and temperature and moisture are factors.
Gotcha. And so one of the questions in the tech or in the chat is how do you classify fungi like plants, like snow plants or pine drops?
Sorry, can you read that again?
How do you classify fungi like plants, like snow plants or pine drops?
Okay. Well, I think you're asking is like how, so classify, you mean like how do I get them to like species? Like how do I identify them? Is that because
No,
fungi
I
have
don't.
different.
sorry, I'll come off mic here. Now, in where I live, we have these plants that are not fungi because they have plant-like structures. But I'm just wondering, how does the fungi community, do you embrace uh, plants that are not true fungi, but are kind of like partial fungi? I don't know how to explain it, but the ones we have are called snow plants or pine drops.
Oh, I think you're talking, those are parasitic plants. We do have some plants that are parasitic on fungi that you can find nearby. And we do have fungi that's endophytic, meaning it like grows in it or sometimes even just on plants. Um, I think we accept anything that's fungal. I mean, even when you think about lichens, lichens are like part algae, part like fungus. So it's really unique, especially with plants, because I think it's something wild. Like 90% of plants have like a relationship with fungi. So it's like if something happens to fungi, then we could lose like a lot of plants. And when you think about fungi in our life, like how much we've benefited from them, like literally from like bread to coffee to like penicillin, like we wouldn't be like we couldn't survive on this planet without fungi. That's how important they are. And it's also kind of shocking that like they are not getting hardly any equal amount of attention as these other kingdoms like plants and birds and etc. I hope that answers your question.
Yes, thank you.
Yeah, and so any other questions out there, put them in the chat if so, or come off mute and ask your question live if you'd like, or raise your hand.
I just
Um,
had a question kind of on the tail of what you just said. So I do work for a restoration organization, and before we break ground on any projects, we have botanical surveys, we have vertebrate surveys, we have like all these different birds, you know. What are your thoughts on fungi? Should we be doing that? Should
Absolutely.
should we be
Yes.
concerned about the other thing that I notice is, once we've come through and we've disturbed an area and there's wood chips or things are masticated, different fungal communities are established there. And I'm just curious, like, what are the things we should be concerned about with
I
fungi?
think fungi should be equal footing. Like if you're putting time and energy into plants, you should be putting the same amount into fungi because in my opinion, they're more important. Like our plants literally can't survive without our fungi. I think doing a survey so you have an inventory or a database of the species on the land you're working on is a really good start, just so you can know what you have and then keep track of it. Is honestly what we were doing. We were just creating this giant database.
Cool, thank you. And if you have, don't want to take up everybody's time, but if you have any resources for, folks in Humboldt who can support us in doing those surveys, please let me know.
Where are you located?
I live in Whitethorn. I work for Sanctuary Forest, and so most of our work is
Oh,
done
that's
in
right.
the headwaters of the Mattole.
I thought you looked familiar. I remember you from the walk.
Yeah, definitely.
Okay,
And
yeah,
we also live right on the top of Paradise Ridge. So there's like, it's the fungal kingdom up here.
yeah.
And I'm just looking, I have access and I want to contribute, but also get support and knowing like what should we be looking for and what should we be looking out for to protect.
I would recommend maybe starting an iNaturalist project for your property. You can create a grid, and well, you already know, like me, Brittany Tonks, Lost Coast Mike. We're pretty knowledgeable and in the area, so like maybe we could come down and do like a survey for you from time to time. I'm sure there's other folks too. It is kind of hard living under capitalism, finding people who are available to volunteer and have the time to volunteer to come down and do these things. And I love that your cat is hanging out for this presentation right now.
That's distracting. Thanks, you guys. Thank you.
So we have another question in the chat. Do some fungi have a relationship with slugs and snails? Food source, symbiotic, parasitic, etc.? Question mark.
They're absolutely a food source. A lot of times when I go out, I take photos of them eating mushrooms. That's really important for our gastropods. I don't know of any off the top of my head, but there probably is some kind of fungus that affects them at some point that maybe parasitizes them, but I don't know of any. That would be an interesting thing. I'm creating a list now of holes to go down later.
And a follow-up question is, are there any non-native or invasive fungi we should be aware of locally?
Absolutely. There's all kinds of non-native species. One that's been in the news lately is the death cap. That's never been in Humboldt until recently. And there's kind of nothing you can do about it. Like once it gets here, mushrooms, some of them have like millions of spores that they release. So like it's like in the soil, it's in the plants. It's like once it's here, it's kind of here. And there's not much you can do about it, which is unfortunate because in these invasive species, they take up habitat for native species that could thrive here.
On behalf of Friends of the Lost Coast and the Bureau of Land Management King Range National Conservation Area, thank you for tuning in to Lost Coast Lectures. To view the complete archive of recordings from this series, or to learn more about our work inspiring a passion for nature, visit Friends of the Lost Coast online at lostcoast.org.